Okay, so let's get started. Hi, I'm Marc. I'm one of the organizers of the MIDOG 2025 challenge, and well, you might know me already from the earlier videos introducing the challenge. And this time I will present to you the second task of the challenge, which is the atypical classification task. So let's get right into it. Before we go into what is atypical, I want to briefly tell you the normal state of mitosis, which is basically then the opposite of the atypical. So mitosis is the process of cell division, which you might already know. And in mitosis, now organizing my screens here. In mitosis, or in order to explain this to you, I want to introduce to you two things in the cell, which are the cell nucleus here, which is kind of the control center of the cell. It stores the DNA, which holds the instructions for all cellular functions. And before the mitosis actually starts, the DNA has already been replicated. The cell also contains two so-called centrosomes, which are those things here, and these help to organize the internal skeleton used to move things around in the later steps. All right, now let's start with the first phase of the mitosis. So the first phase is called the so-called prophase. And in the prophase, the copied DNA condenses into two distinct units called the chromosomes. And each are made up of two identical strands of so-called sister chromatids. Now, the structures called spindle fibers, they start forming, and these are like cables that will pull the chromosomes apart. The centrosomes then move to the opposite sides of the cell. Then we go to the next phase of mitosis, which is the prometaphase. In the prometaphase, the nuclear envelope, which is the membrane around the nucleus, it breaks down, giving the spindle fibers access to the chromosomes. Now, each chromosome gets connected to spindle fibers via a small connector region called the kinetochore. Right. So that's the prometaphase, and that's the phase preceding the so-called metaphase, as it's also sensible. In the metaphase, the chromosomes line up in the middle of the cell. Think of it like staging them in the center before a dispatch, and each sister chromatid is connected to opposite sides of the cell by those spindle fibers. Now, of this we have here the same situation, but this time it's not the cross-sectional view, but it's viewed from the top. And as you can see on the right, this forms a different pattern also in the H&E, which more closely resembles a ring shape. Now, let's go back to the cross-sectional view. It's the same phenomenon, just viewed differently. It's a 3D phenomenon in the cell. So yes, this is like two ways of looking at the same thing. Now, in the next step, click here. So in the next step, which is the anaphase, the connections between the sister chromatids are cut. And then the spindle fibers pull each of these halves towards the opposite ends of the cell, and by this they're separating the genetic copies. Now, after this is done, we're going to the next phase, which is the telophase. And in the telophase, we basically have new control centers that are forming. So the chromosomes unpack, and we have new nuclear envelopes around them. Then the internal transport system, the spindle, is dismantled, and basically now it's the step that is then preceding the final step. The final step is the so-called cytokinesis, and in the cytokinesis, the two cells split their contents and membranes down in the middle, and creating two separate daughter cells, and each with a complete copy of the original DNA. So that's the normal process of mitosis. Now, the interesting question is if there's a normal case, what are the not normal cases, the so-called atypical cases? So our little cell here is maybe feeling a bit atypical today. So let's have a look at how that looks actually. So there's different cases of atypical mitosis, and one is a so-called polar asymmetry. And in the polar asymmetry, as you can see, not all of the chromosomes have been, or not all of the sister chromatids have been pulled to the right sides, thus indicating a mismatch of those DNA available to all of to the new daughter cells. So you have on the one hand one daughter cell that doesn't have enough DNA, whereas the other has too much DNA, which could also lead to misfunction. Then we have another case, which is so-called a tripolar case. In the tripolar case, we have actually multiple centrosomes building multiple centers, building multiple of those metaplane axes, metaplate axes. This is of course also not intentional and does not, since we only have like 2 sister chromatids, it cannot lead to 3 fully functional cells. So this is also dysfunctional. Finally, as a final example, at least, because there's many more of those divergencies here, we have an effect called lagging chromosomes. Lagging chromosomes are like visible chromosomes that have not been pulled to either of the sides and are thus also missing in the new daughter cells. So this is very briefly the process of atypical mitosis, and as you can see in this image, well, there's like so many different ways an atypical mitosis can actually be seen in a histopathology sample. So one of the challenges in this task is actually that atypical mitosis is highly diverse. So we have a high diversity and a high interclass difference here. Okay, so let's go ahead into the next. So this is the challenge that we have in our track 2 of the MIDOG challenge, and we want to thus have a binary classification between normal, here depicted on the left hand side, and atypical, here depicted on the right hand side, mitosis. Alright, why is this important now, you might ask yourself. Well, as with the normal identification and counting of mitosis, of course, it also has to do with tumor malignancy here. This is a fairly recent development that we know about this. I just have like 3 papers basically indicating this. The first 2 showed this in breast cancer. The third one, I have to admit, I have to look it up again, but it was for sure some veterinary sample because it's from my colleague and co-organizer of the challenge, Christoph Bertram. And as you can see in those Kaplan-Meier curves, basically we have a strong prognostic relevance in the overall survival of patients. So the amount and presence of atypical mitotic figures is a prognosticator, and it actually has been also shown to be an independent prognosticator for survival. Now, much more recently, a group around Hannefer from Warwick also showed that this is not only the case for like the breast cancer cases, but also for many other cases. So this has been been put out, like I think 2 days ago on medRxiv, and it's a very interesting large-scale study also showing the importance of atypical mitosis. So I think this should give us all ample motivation for this task and for solving this task as a, well, computer vision challenge. So let's now go towards the challenge setup. So let's start, and I want to reveal some details. So first of all, what is important to know is that, opposite to like many other challenges, we do not reveal any test images. So we just don't want participants to have access to the test images as it would allow overfitting to those test images, because this challenge is also strongly about robustness, and we don't want that to happen. So the participants instead have to do a Docker-based submission where the Docker container will then get access to the test images at runtime. Then what does the container have to process? Well, it basically gets images, most likely a batch of images of those crop outs centered around mitosis. This is always a kind of validated mitosis in a 3-expert consensus, and these mitoses will then have to be classified into being normal or atypical. Furthermore, we also want to have the confidence of each classification given by the model. Right, what datasets do we provide? We provide actually 2 datasets for this. One is a little bit older, though not old, it's also from the same year. It's the so-called MEVR dataset. It's a dataset that is consisting of cases of atypical and normal mitosis in breast cancer. It has been sourced from the MIDOG 21 dataset and from the 2PAC16 dataset, so it's all breast cancer cases. We have a 3-annotator blinded majority vote on if that is like atypical or normal mitosis, and in total it's like 3,720 cases that we have. Then we have very recently just released the training set of track number 2, which is the official MIDOG 25 training set for track 2. You can find this on Zenodo and on Google Drive and on wherever else we published it. You can find it on our webpage. And it consists mitotic figures from 7 tumor types, also including human breast cancer, but not exclusive to that. So we have several kinds of human and veterinary cancer types in there. It has been sourced from the MIDOG++ dataset, which actually also includes the MIDOG 21 dataset. So there's a little kind of dataset bleed in between those. Make sure you don't fall into that trap when handling them as independent sets for, like training and holdout sets. In total, it contains 11,937 individual mitoses, and we also have a 3-annotator blinded majority vote on them being atypical or normal. All right, I think that's it for now. What's next? The next steps will be, well, first of all, we are currently working on a Docker container with a reference algorithm. So basically showcasing you how we want an algorithm to be included into a Docker container so it can run on the Grand Challenge platform. Yeah, that's also some news. We are using, as in previous years, the Grand Challenge platform for inference on this and for basically equal opportunities for everyone. Then on August 15, we will have the submission enabled on the preliminary evaluation set. So please make sure to register. You can already register right now, and then you can also talk to other people that are participating in the challenge in the forum. You can also do that in our Discord, of course. That's that. What else to say about that? Yeah, there is a so-called preliminary evaluation set, as we did it in MIDOG 21 and 22. One thing that you have to absolutely know about this is that this evaluation set does not share a lot of, dataset properties with the final test set that we then run for the final evaluation of the challenge. Why is that? Now, we've seen this in the past actually with MIDOG 21, that then people tried to kind of overfit on the preliminary set. Since in MIDOG 21 the preliminary set had at least some it was different cases and so on, but it still had some shared properties with the final test set. It was not like completely independent in that sense. This was successful, and this is not what we want in a challenge, right? So we want to see generalization of algorithms and not if people are able to overfit on some dataset characteristics. So here again, we will have a completely disjoint dataset, not sharing properties with any of the test set. So make sure to not overfit on that. I know it is sometimes the thing to do, right? If you get a better score and you get a better score and you better get a score by iteratively trying on that evaluation set because you have multiple runs on that set, but I would advise you to not do this. So rather optimize with your own means until you feel satisfied and use this only for technical evaluation. And then finally, on August 30 and 31st, we will have the submission on the test set. Well, and then of course following the MICCAI workshop in autumn. Right, that's it for now from my side. I hope this was interesting to you. I hope that you are now much more motivated to become an active part in our challenge, and then, well, see you there. Bye-bye.